Hey everyone, and welcome. Today we're going to do something that sounds super high-tech, like it belongs in some fancy lab, but you can actually do it right in your own kitchen. We are going to pull the literal blueprint of life, DNA, out of a strawberry. And you know what the best part is? You probably have everything you need to do it right now. So, let's get into it.
Just think about that for a second. We're about to isolate and actually see the molecule that holds all the genetic instructions for a living thing. It totally feels like magic, but I promise you, it is 100% pure science.
So here's our game plan. First, we'll look at why a strawberry is the perfect victim for our experiment. Then, we'll walk through the step-by-step how-to. After that, we're going to break down the science behind why it all works. And finally, we'll see how this simple little kitchen experiment connects to some world-changing medicine.
All right, let's kick things off with the star of our show. Now, you could use other fruits, but there's a really specific, kind of nerdy reason why strawberries are an absolute rock star for pulling out DNA. So, what do you think? Any guesses? Why this specific berry? It's not just because they're super easy to mash up, though, that definitely helps. The real answer is hidden deep, deep inside its cells.
And here is the secret weapon. Strawberries are what's called polyploid. Specifically, they're octoploid. Now, we humans, we get 2 sets of chromosomes in our cells. A strawberry gets a whopping 8 sets. That means every single strawberry cell is just bursting at the seams with DNA, which makes it way, way easier for us to see.
And hey, don't just take my word for it. Check out this data. Somebody actually put this to the test, and the results are crystal clear. Strawberries, our octoploid buddies, give us way more DNA than kiwis or bananas. It's really that simple. More chromosomes equals more DNA for us to grab.
Okay, enough theory. It's time to roll up our sleeves and get our hands a little messy. Let's move on and build our very own at-home lab so we can get to the good stuff.
Okay, here's your equipment list, and I'm betting you have pretty much all of this kicking around your kitchen already. We need some strawberries, a Ziploc-style bag, a little dish soap and salt, water, a coffee filter, a clear glass, and some cold rubbing alcohol. That's it. Science doesn't get much more accessible than this.
So step 1 is simple, and honestly, it's pretty fun. Just pop a couple of strawberries into that sealable bag, squeeze the air out, and then just mash them up. Go for it, for about 2 minutes. You've just started the process of breaking open millions and millions of tiny plant cells. And this right here shows you exactly what's going on. All that mashing is physically tearing apart the tough, rigid cell walls that act like armor for each plant cell. We've got to get through that first layer of defense to get to the treasure inside.
Okay, time to mix our magic potion. In a separate cup, just combine about a half cup of water, a teaspoon of salt, and 2 teaspoons of regular dish soap. Give it a gentle little stir. This simple mix is actually a really powerful chemical tool.
Now, I know this looks like just some soapy, salty water, but trust me, each of these ingredients has a really critical job to do. This is our extraction liquid, and it's what's going to chemically bust open the rest of the cells so we can get to the DNA. We'll talk more about how in just a minute.
All right, go ahead and pour your extraction liquid right into the bag with all that strawberry mush. Seal it up, and then just gently squish it around for about a minute. Next, you'll set up your coffee filter over a clear glass, and carefully pour the whole gooey mixture in. Then, just let it drip. So what's the point of this step? It's all about separation. The coffee filter is catching all the big stuff. The seeds, the pulp, all the broken cell chunks we don't want. But it's letting the liquid that has all our dissolved DNA pass right on through.
Now, before we get to the final, and frankly, the most exciting step, let's hit pause. Let's actually understand what we just did. Why did we mash it? What's with the soap and salt? Come on, let's break down the science.
First off, the mashing. This has a fancy name, mechanical lysis. Lysis is just a sciencey word for breaking open. By squishing those strawberries, we physically busted through that tough outer cell wall, which is kind of like the first security gate. Next up, the dish soap. That performed what we call chemical lysis. See, cell membranes and the little membrane protecting the DNA inside the nucleus are made of fats. And what does soap do to grease on your dishes? It breaks it apart. The soap does the exact same thing here, dissolving those fatty membranes and letting the DNA spill out.
And the salt? What was that for? Well, its job is to help the DNA precipitate, which basically means to make it solid so it comes out of the liquid. The salt helps all those long, stringy DNA molecules, which are normally invisible, to start clumping together, forming a big enough blob for us to actually see with our own eyes. All right, Back to the lab for the grand finale. This is the moment we've been waiting for. Take your cold rubbing alcohol, and it really helps if it's cold, and very, very gently pour it down the side of the glass. You want to create a whole separate layer right on top of the red strawberry liquid. And whatever you do, do not stir it.
And now, just watch. You don't have to wait long. Within seconds, you'll start to see it appear. That white, cloudy, kind of stringy stuff that's forming in that top alcohol layer, that's it. That is strawberry DNA. You literally just pulled the code of life right out of a fruit.
How cool is this? See, DNA can't dissolve in alcohol, so when it hits that layer, it all clumps together and becomes visible. And now you can actually take a toothpick or a little skewer, dip it in, and gently spool the very blueprint of life right out of the glass.
So we've got a gooey glob of DNA from a strawberry. Pretty cool, right? But what's the big picture? Why does this fun little procedure actually matter in the real world?
Well, because what we just did in our kitchens, that's the absolute critical first step for so much of modern science and medicine. When scientists needed to create vaccines for COVID-19, they started by extracting the virus's genetic material. When we mass-produce life-saving insulin for people with diabetes, that process starts with DNA extraction. It all begins with these exact same principles.
So now it is your turn to be the scientist. What happens if you use a banana instead? Or more salt? What if the alcohol isn't cold? Remember, only change one variable at a time. Challenge your friends, play around with the protocol, and see who can get the most DNA. Go find out what you can discover.